let's go.
There you go.
All right, we're good. And going to meet Philip. And yes, Frank, so thank you for taking the time to be with us. And if you can then just share a bit of the kind of intellectual journey, spiritual journey brought you to the point of your presentation, then go into your discussion. And then we'll have time afterwards to further discuss and ask questions. I think it's important just to understand, Frank, how you've come to where you are now and some of the important lessons that can be learned. So Frank, I'm going to turn it over to you, and I see Vishal is joining us as well. So we have a distinguished audience here.
Great. Yeah. Can I show my presentation? Just
yes, you may indeed. I'll just keep an eye on the waiting room. And you've got screen share, so please do.
How do you get volume on there? How do you get volume? Well, it's these two outside ones. What's happening here? No, no, nothing's happening.
There we go. Okay, Frank, we're in your hands here now, and I'm going to keep admitting people as we go. But I'll turn it over to you. And again, thank you for taking the time. And I'm assuming some of this will be available afterwards. At least I'll try to record, but if the slides are available, that would be helpful too. Okay.
Okay, just shortly. I was born again, as one say, filled by the Holy Spirit when I was 13 years old, and was completely transformed at that moment. Born by blood, by Christ's blood, and saved to the bone, as such. So that's many years ago, and have been a wonderful life, kind of saying. And I have been studying heavily the Bible for so many years, and all kinds of things related to the Bible. But my background such is that I have a master in chemistry and biotech. I've practiced in clinical bacteriology. I have a Ph.D. in molecular biology, virology, and pathology. I have a postdoc in so-called environmental microbiology related to water, drinking water technology. I've been head for the research in some commercial issues for 10 years. And then finally, I was accepted first time as a professor in 2006, and then also a second time in 2010 by an international panel of professors who accepted me as a professor in a lot of disciplines. You can talk about nanotechnology or lab on a chip, and I have 20 international publications here, also 28 patents behind me.
You've been busy.
I've been busy, and so during all these studies, what I did, together with studying the Bible and learning more from heaven, I discovered more and more that the Bible was very much true. It had been a long journey, but I've always believed 100% in the Bible, and I'm still in. And it just been revealed more, I think, by the Holy Spirit, things in the Bible that I've never seen before, and it's kind of everyday revelation to me. A lot of things that is happening. So that was the short startup.
Right. I have actually a question, which is totally gründer. You were the gründer. What is that? What's the English translation? Or what's that equivalent to?
And it means somebody starting up a company.
Okay. Oh, founder, I guess, really.
Yeah, founder. I've founded many companies. I don't know the numbers. I think it's 20, 30.
Okay.
Up to the years.
That means founder.
Is everyone muted? I think we are, but just if you aren't, please mute yourself while we're in the discussion, or Frank's presentation, and then unmute when you want to discuss later.
Just a wordsmith here. The gründer is out of a Norwegian word, and that has more the innovative part. The founder is more you're founding a company, but
Okay.
here it's the innovation part, which is more carrying the one.
Okay.
So,
Thank you, Eric. Yeah, I wondered. I looked it up. I didn't really get very far, so that's a good. It's an important term, clearly.
Okay. Do you want me to go ahead and?
Yes, please. Yes, please, Frank.
Okay. I'll go ahead then. Let me see. Can you see it?
Yep, I think everyone sees it. Looks
This-
good. Yep.
You
full
see the
screen.
bio profile now?
Yeah.
Yeah.
That's
So
good.
just to start from the beginning here, and to be very 100% sure,
Right.
it was God in person that created. And for me, that is science more than anything else. A person has to be involved. For me to do science or laboratory work or research, whatever, without having a connection with a personal God is kind of foolishness. Foolishness doesn't work for me. So, and God the Father is the Father of Jesus Christ, and they are the one who created, and he's defined as God of Abraham, Isaac, and Jacob. And so there was no undefined God that was creating. It was him, the Father of Jesus. So I'm going to focus on the Genesis, the first part here. And I'm talking about Genesis chapter 1, verse 1 to 2. Now the earth was formless, and in the beginning God created the heaven and earth. And now the earth was formless and empty, and darkness was over the surface of the deep, and the Spirit of God was hovering over the waters. This is kind of, for me, starting the events of the 6 days, and not a minute more of creation. He created within 6 days. He did it out of nothing, and that is also emphasized in 2 other places in the Bible very clearly, or in Exodus 20 and in Exodus 31. For in 6 days the Lord made the heavens and the earth. So there is strong biblical evidence for that. So there is no reason to believe otherwise. It has to be 6 days, and God wanted it to be 6 days. He could have done it maybe a longer time, but he chose to do it within 6 days. I'm sorry, I'm a Norwegian, so my English is not 100% perfect, but I hope it's okay.
Good.
So I was the first one who probably created an accurate pre-cancer diagnostic method, and some of you have heard about it before. Inspired by HIV and bacteriophage T7, and it is still the most accurate pre-cancer diagnostic method there is. And there is nobody even close worldwide that have anything related to it yet. But I'm going further. I've done a lot of other things. So we are bringing us quickly into the kitchen, where we are thinking about doing disease diagnosis in the near future. And that's my technology these days. It's called Salico. It means health control. And we want to bring the highest possible accuracy of diagnosis into any kitchen worldwide. And at the same time, it's going to also happen inside the bathroom, that you can do all kinds of diagnosis. I'm just giving a short introduction here before we go on for day 1. So this is just showing very quickly the first scenario of worldwide primary screening system that can be done back at home by women themselves. They can take the samples at home, a self-collecting device. They can test themselves back at home, and they can the results is digitally sent to a medical doctor, that can recommend then to remove the problem, and the woman will not get cervical precancer at all. So that's part of the very interesting technology going on now, which is in the commercial phase, and we are conducting test production these days to cover this kind of disease, but also many other kinds of disease. Let's say the SARS-CoV-2, the HIV, tuberculosis, malaria, and so on. But let's jump into the point. God created or established a space and an object so that he had a place where he could create. And that's the point. I mean, God couldn't start without any space. He started, of course, with a space and an object. The object was, for me, the earth. And the project that he started here is the project, and it's no meaning without having a start. He has to start. So the project must have always had a start date. So everything started, the time started, as far as I know. So it was completely empty and clean, as we already read. It was empty and clean. And for me, this is a room, a place which have to be 100% clean in the beginning. So I don't think God or Elohim, God the Father and the Son and the Holy Spirit, started to create in a mass. It must have been a clean area. And I'm coming to that. I mean, we are not talking about an unclean area. So this is a nice little kitchen, but I don't think anybody would like to make any kind of food there in this room. So you start with a clean space, a clean area. So here we are talking about day one, where God has everything in a clean, and you can compare that with a clean laboratory and an empty space, and where you have control of everything, all the steps. So God has this earth and the space around it where he could start and do the things. And there was no interfering particles or random single molecules there. It couldn't have been. And that could completely prevent the molecular biological reaction. So of course, God, he started with something very specific and very clean. So God created all this in the beginning, the pure molecules, and he did it. He did all the variants, but only the molecules that was completely necessary in the biological system. And of course, you don't have a laboratory with nobody on board. You need somebody there to work. And this is some of my students, and they have to work hard. They need all the tools. They need all the tools they need to have there. It has to be completely pure. You know, you have to work with the right concentrations. It has to be highly organized, and it has to be highly managed, and it has to be an optimal plan behind everything that you do in the lab. So I compare this work in the lab with what God did in the beginning. And I strongly believe that when you say that the earth was formless, without disturbances, an empty darkness with right conditions was over the surface of the deep, and the Spirit of God was hovering over the waters. I think that means we had a deep, very pure water covering the whole earth. Very deep and just water. Nothing disturbing there. And the space, the heaven around it was at the right pressure, with the right temperature, without disturbing particles. If you had the pressure of all this, which you can see in the atmosphere today, is that there is 1,200 degrees of temperature. When you go out in the outer space, you'll find temperatures which is not useful in relation to a creation. So it had to be the right temperature, the right pressure, without disturbing chemicals, without disturbing particles. Is this clear, or is there anybody asking a question here? No?
Good.
It's good? Okay.
Excellent.
Excellent. So the light was then created. God saw that the light was good, and he separated the light from the darkness. And God called the light day, and the darkness he called night. And there was so. There was evening, and there was morning the first day. And for me, this clear light is nobody else than Jesus. As you can see, he was there in the beginning, and he was in the end. Jesus has always been there. So for me, this is the introduction of Jesus Christ himself, the Son of God, the whole picture of Elohim, the Father, the Son, and the Holy Spirit. I mean, compared again very quickly, he didn't make the march. The march was completely impossible. And you know, the dream of going to Mars is completely ridiculous because there's nothing there. It's just hopeless environment, and there's nothing there which attracts anybody or any human. It's not made for humans. It's not dedicated for humans. Everything is against doing anything on Mars. Therefore, I launched my own idea, Make Sahara Green. Is a better idea and a better project. And just after the flood, you can see how the Sahara looks like, and it's already evidenced in a new paper coming out by the King's College in London, where you have the mega lakes. So I recommend all of you to read this paper. It's a fantastic paper, and you can see the evidence of how it looks like after the flood, just after the flood. But again, when God started the movement, I think he started to make the space around the earth. There is so marvelous things that can be done here. As a chemist, I would love to go deeper into all these layers of atmospheres around the earth. And we have one with us, Philip Panic. He knows, of course, this better than I, but there is so much things that haven't been studied yet, and which, if you base your studies, if you base your science on the Bible, there is so much interesting stuff to be studied. How God did this within the first days, and within the first and second day even. How he did it, and how he created everything in the relation to the pressure and temperature that has to happen within a very short time. Otherwise, nothing would have been able to survive in the lowest area of the atmosphere. So the multibacteriation process had to be done within 6 days, as the Father has already decided. So there have to be some sort of planning, and we have heard about. There had to be some sort of planning. They planned it together before they started the creation. Obviously, there is. I mean, the design. Of course, they have designed. But design is not only the picture. There have to be calculations, measuring, modeling, verifications, constructions. So God created with his word out of nothing, but not without a very intelligent hard work. So the Father did a very hard work done by the Father, the Son, and the Holy Spirit, including a super planning, a super measuring, super modeling, super verification of the organization, construction, assembling, systematization, validation. So the creation, to my point of view, demand right timeline, right design, right mechanics, right chemistry, right tribology, right pressure. You see my point?
Yep.
Right combination, right components. You see my points?
Right.
So in the first day, we start to see a lot of interesting subjects that is covered within just the first verses here, that you have a lot of interesting scientific subjects, and there is even many more new subjects that is coming up. So the scientific subjects is covered within inorganic chemistry, particle physics, cosmology, planetology, science, meteorology, and so on. And there is thousands and thousands of papers that would help us here to see how God did it. God make a plan, God make a salvation plan, of course, the salvation plan to the cross. The three persons had to accept the plan, all of them, by their own free will. The three persons of God had to accept their roles in the plan for the salvation. The plan was initiated on day one of creation, and Jesus Christ was announced to be the light of the world on day one. So that means that the time began to run. So for me, the plan for salvation on how did God this, Jesus had to die exactly this Friday, 5th at 3 o'clock or 3 p.m. in the afternoon, 4,000 years after the creation. And that was the key thing. So, I mean, we can see that it was extremely difficult to be able to see that everything came in place in relation to social anthropology, in relation to mathematics, in relation to political processes, and so on. So there have been a lot of things that had to be extremely clearly planned. So back to the point is that the seventh day, God finished the work he has done. So on the seventh day, God completed the work. So now we are talking about the work that the Elohim, the Adonai, did within these days. And it was completely planned, very much planned to the little point of everything what he did. So I think that there was a plan behind it. And of course, it's all about Jesus Christ died on the cross. He stands above all his creation, and in him all things have been created in heaven and on earth, visible and invisible. Everything is created by him, for him. He's before all things, and everything is made by him. So through him, all things are reconciled to God our Father, all things in heaven and on earth. So this is all what you know very well, all of you. And so I believe that we can preach on the honor of God the Father and his Son, Jesus Christ. Everything came out within these 6 days. So there is also a lot of new subjects that can be mentioned in this process of creation, and we can come up with many new subjects that we can call whatever we like. So many new subjects can come up, and new kind of institutes or departments in universities can be created based on these new subjects that we can learn from this 6 days of creation. So I'm now quickly jumping to the beginning of what I wanted to talk about. My thesis is related to that the time is the curse for the idea of evolution. It makes evolution not possible. So the time, when you see these 4 compounds here, it doesn't help giving it time. All this had to happen as quickly as possible, moving all these compounds together and mix them as quickly as possible in a pure condition, and to use a lot of time on this process or this work is not very useful. So again, we come back to the day 2. Let there be a vault between the waters to separate water from waters. So God made the vault and separated the water under the vault from the water above. And it was so. God called the vault sky, and there was evening, and there was morning the second day. So on the second day, the atmosphere was created. So this had to create the possibility of giving people a very much longer life. So the humans in the beginning, they had an atmosphere that was ideal for very long life. So there's a lot more that can be studied on this, but humidity may have been more perfect, the temperature better all over the world. and there could be a sharp reduction in the radiation from everywhere, and so the perfect climate was established everywhere. And the magnetic field was maybe stronger than today, created a better atmosphere. And also, it's very clear that this kind of picture, when they say, I took this from the University of California, Department of Medicine, who wrote this paper recently, and they talk, of course, about 4 to 5 billion years. But they say, if you read in the middle there, you can see this was due to the miracle of photosynthesis, which enables some organisms to use the energy in sunlight to break down into its components, molecules, oxygen and hydrogen. And here you see, they say it in a scientific paper, they call it a miracle. I can read these miracles in so many papers. I think you can find this word miracle in so many biological papers you can't even imagine. I mean, I think we can collect thousands of papers where they mention the word miracle. And so they created the cyanobacteria here, which they say is 2 billion years, of course not. God has to put that in place within 6 days in order to keep this exact gaseous situation in the atmosphere. But the concentration had to be exactly 78,084 ppm. And the oxygen had to be 20.946%. And of course, this perfect situation had to happen with these ideal gases there, argon, neon, helium, and this had to be there in order to keep this temperature. So God has to put this in place very quickly in order for anything to survive. So this couldn't be, I mean, you have heard about so-called bacteria that can survive just without all this. It's not true. There is no bacteria that can survive alone or in a small little homogeneous population. They have to be together with hundreds of other organisms and to play together with them. So this had to come in place very quickly within minutes after the creation. So in day 2, God has to put all this in place, as well as the perfect salinity, which can find, it means that the salt concentration is variable, but the salt, the salinity within the salt concentration is stable, and it's always been stable, and God secured that. And that is no variation, like with this one is staying constant, as well as this one, showing that God just put everything in perfect condition for what's going on in the 3rd day. And God said, let the water and the sky be gathered to place. And again, all these kind of things had to happen very quickly within minutes. You can't just wait for this kind of thing to happen. It had to happen within minutes. So there's don't talk about hours. Don't talk about weeks or years. Yeah, that's ridiculous. It's nothing to do with science. It had to happen within minutes. So the 3rd day, he said, and God said, let the water and the sky be gathered to one place and let the dry ground appear. And it was so God called the dry land and gathered waters he called seas, and God saw that it was good. 2nd day, he didn't say it was good. It's because he wanted it a better situation after the flood of Noah. Then God said, let the land produce vegetation, seed-bearing plants and trees on the land that bear fruit with seed in, according to their various kinds. It was so. The land produced vegetation, plants bear seed in it according to their kinds, and trees bearing fruits with seed in it according to this kind. And God saw that it was good. And there was evening and there was morning the 3rd day. So we can then look at the day 3 as splitting up into the atmosphere of what was going on in the atmosphere to secure that all the cycles of all kinds of chemicals was in place in the right condition in the atmosphere. Also on the different layers of the atmosphere had to be in a perfect condition before you ever can think about planting, having any plants placed. And you couldn't stay that stable condition in the atmosphere unless you had a perfect production in the ocean and in the ground, and as well as on the ground. So you need all the chemicals within the ground that had to be there in the right concentration with the right purity, produced by the right organisms at within very short time. And the same happened in the ocean. With what I just said, it's the Protochlorococcus, which is the bacteria you can see on the right-hand side there, which is producing maybe up to 70% of the oxygen that we breathe in, and it produces much more than the whole tropical forest. In the beginning, the diversity of the phytoplanktons was clear here, and there are many new papers coming up with this discovery that this world of which we call cyanobacteria or phytoplanktons is in fact keeping everything in perfect balance. So the Jesus and the Holy Spirit had plenty to be done within the third day in order to start this cycle here, for instance, sulfur cycle process. And there is 7,497 papers, scientific papers that confirm this. And it is pretty clear also that you had the same with the nitrogen. You need the right condition in the ocean, and the cycle had to be in place for everything to work on for preparing the fifth day. And again, we see the cycle of combinations here between the carbon cycle, sulfur and nitrogen. It's kind of completely dependent balance between these. And they talk about carbon dioxide. Well, there is 20 other compounds with carbon in that had also to be involved. So it's so stupid to talk about just carbon dioxide these days.
Right.
Yeah. Is there anybody who wants to interrupt? Any questions? Am I going too quick?
There's a lot there. You mentioned a bunch of papers that are already have really confirmed a lot of this discussion, and perhaps no one's ever pulled it all together, I think, in the way that you have. So that's very fascinating, and definitely would like to follow up on some of those references as well. But I think your point about the fact of the intricate fit of everything, and it's not just, everything has to be just right for the kind of process that we now see in terms of life being formed. So I think this level of analysis or discussion is very, very insightful, and generally tends to be sloughed off or just talked about as a miracle when it's not explainable by the naturalistic approach.
Exactly. I mean, this is showing that there was a hard work of the Godhead, and it had to happen. Also, it's also shown that they have to happen within minutes and not days or weeks. But again, also when you have to make the tree start, you couldn't have baby trees the first day. You had to have adult trees the day three. You had to have all the things in the ground had to come in place with all the bacteria and yeast and fungus that had to be there in order to produce pure nutrients in place for the full mature tree to stand there, and millions of trees, maybe tens of billions of trees had to be in place at the same place all over the earth at the same place, and the same in the ocean. You had to have the ocean ready because we are coming close to day 5. And everything had to be in place. All the nutrients had to be there. And nothing can just go gradually in a gradual process. It's completely impossible in relation to science. You had to have everything in place, and all the work had to be done within as short time as possible. So when we come down to the sun and the sun part, you know about it, and the stars is just mentioned like something, as nothing in particular important. And the point was that you couldn't have had the sun and the stars, to my point of view, you couldn't have had the sun and the stars there in the beginning. I mean, before the day one, you couldn't. It's not possible. You couldn't have any stars or any sun on anything the 3 first days. That would create a lot of trouble for the creation. So it's very clear that it has to come in place on day 4. And by the way, the stars is not very complex, but the cell, which is in our body, is much more complex than a star. And everybody's saying that the galaxies and everything out there is so fantastic and blah blah. They haven't seen how fantastically the biological world is. So summing up the 4 days, enough organisms and nutrients had to be produced in the atmosphere, soil, and sea for optimal plant production. So gases in the atmosphere and the ocean are stabilized, and you have enough oxygen, you have enough of everything in order to create the right pressure and so on. And the day 5, we know that it comes into place, the whole ocean and the birds. And the complexity is huge, as you know. It's fascinating to see. There is so much to be studied, and there is no other meaning that there is an ultimate personal superpower involved that has created everything. And just even the membrane of a bacteria is extremely complex. So I'm just pushing through here now. I'm coming to my point now, which is more important. I'm just giving you a summary now of the 6 days first, and you can see how fascinating is birds can fly so fast, up to 153 kilometers per hour. I mean, how can that be? But that is what is going on. So on the 6 days, you get the most ultimate creations on the whole thing, and you have in our body we have the number as you see here. 37 trillion cells. And how on earth has then been able to make all the cells at the same time in the same system? So of course everything is interdependent, and it had to happen within the same in the same time as God said, and it happened. So all this together, all the system, all the processes internally in the organism had to come in place very rapidly, and it is completely impossible to see anything related to a gradual process at all. So as you know, we look very different, and that's fantastic. So the symbiosis in our body had to come in place, otherwise we just would have died. The toxins of just eating a little bit food would kill us very quickly if we didn't have a complete symbiosis. And the last articles on this shows that we have more than 3,000 different kinds of bacteria, and millions of them, but different kinds of. But then I jump quickly into the main point of my kind of new understanding of this 6 days creation. And you know, they love to start with their first ancestor cell, you know that? They love to talk about the so-called first protozoa, the first histolytica, and they claim to say that that is a simple little cell. You have to make it so simple and then gradually grow up to a more complex cell. That's not true. It's nothing that is simple in this kind of story here. To build a protozoa is completely impossible today, and we just understand a little bit about it, that it's extremely difficult situation, and that's quite clear. So I want just to now jump into the complexity of this. Just first talking about the membrane surrounding these cells, you know, and this membrane, just to build it up, is extremely difficult process, but it had to happen while you are seeing it. If you want to get a live single cell working, all of this membrane part, all over the cell area had to be in place and had to work at the same time, at the same second, within the same minutes. So talking about the gradual process is completely radical and has nothing to do with science, or nothing to do, at least, with the laboratory work I have learned. So natural molecular biological reaction can only occur within cells and bacteria. It cannot happen outside of a cells and bacteria. So you definitely have to have the cells in place first. So no molecular biological reaction can take place within a functional, without a functional cell. No molecular biological reaction can happen at all. It's only inside a cell that the purity is good enough. You cannot find anywhere in the nature where the purity is good enough. Not at all, not even close. So to talk about 3-4 billion years ago that there was a situation there which was good enough, not at all, not even close. So it's only inside a cell that chemical concentration can be adjusted exactly. So it's only inside a cell that replication, for instance, the most advanced parts of replication, transcription, translation can take place. And it's only inside a cell that DNA, RNA, and proteins and advanced proteins can be made. You cannot make anything of that outside of a cell, whatever you dream about, whatever experiments you think, it will never happen. It's impossible. And all of us knows that very clear. All dealing with molecular biology know it's impossible to make anything outside of a cell. So also inside a cell, the distances are short enough to be able to see how that is going to work. And by the way, all the cells in a mature body of an eukaryote or a functional body had to be in place at the same time. I'm coming back to that. So the building blocks for the DNA and RNA and phospholipids, I mean the membrane and the proteins, all of that has to be produced inside a cell. So a complete functional cell had to be in place before you could see anything of this going on at the same time. So then I'm coming to the key point, the building blocks. Here you see amino acid, tryptophan, for instance. How to make this molecule here? There is nothing in the nature who can make that by itself. There is no chemistry that can just happen and then it comes by itself. It will never happen. The concentration will be not right. The purity will not be right. So God, he made this within seconds and he made it in place. He built 24 of them. And they are very different and very advanced. And as you can see here, you have to have in place nitrogen, oxygen, carbon, and hydrogen. And where did that come from in the pure enough concentrations? Yes, it came from the atmosphere, which God already had made. But it had to happen within short time. The concentration would dramatically be changed if you didn't do it within seconds or minutes. You see my point? So when you go, so that's the building blocks of the proteins. But then when you go to the building blocks of the DNA or RNA, you can see the same problem here. Is the building blocks become even more complex. And how on earth can you make this? You need a very advanced industry or company or production in order to make this kind of things. There's very few companies in the world who is able to do it yet. But again, the concentration in nature have to be exactly correct, and you can't make it out of the cells. So the building blocks, there's a cytosine, and you need 5 of them in order to build DNA and RNA. So everything has to be made in a pure condition and with the right concentration. You see my point? So here again, we can't leave this for a gradual process at all. It's not possible. The only chance you have in order to build something of a DNA or RNA or protein, you have to be done this rapidly within short time. So talking about millions of years, it's just ridiculous because it would never help anybody. it would be just stupidity to my point of view. So here again, when you look at the building system within the membranes of this so-called first ancestor cells, they are clearly extremely complex. And again, you say the same problem again. How on earth are you going to create the right purity and the right concentration out of this in order to build this very advanced membrane around all the cells or all the bacteria that we have? And the concentrations, and there are so many of them. There is not only one kind of phospholipids. There are all kinds of different ones of them, and they are very complex. Am I running too quick now, or
It's
is
clear.
that okay? You catch my point, or am I talking just what you understand? What you are with me here?
Yep, no, it's very compelling as you present it.
Yeah, because I don't understand how this purity or concentration or anything like this can come into its place within a long time. I don't understand how this can happen without an extremely intelligent work. I don't understand how this can happen within millions of years by nothing. So again, this is, behind this process here, which I've talked about now, of making membranes, of making the DNA building blocks,
yep,
of making this amino acids. There is millions of papers behind it to say that you cannot build anything without the right purity. I can build millions of papers behind this argument.
Right. Now, I just see Warren and Tessa have their hands up for a question. Warren, you go first, and then Tessa, perhaps.
Let me stop there. It is good to
Tessa had her hand up first.
Okay, Tessa, ladies first. Yeah.
Thanks. I had a question back several slides where you had that the concentration of salt, I think it was, in the ocean, and it was dependent on decay. And my question is, how did you arrive at the idea that there was death before the fall?
Oh. Well, that's another question.
You didn't deal with that.
You know,
Frank.
I'm not talking at all about. I'm just presenting my theory now on the Genesis chapter 1, and of course, the point of being death before the sin came into the world is, for me, far from any science that I can talk about. I mean, how on earth can you have death before? Because when God make everything perfect, when everything is good and very good, why are we talking about death?
Well, that was my question, is that it looked to me from the slide like the salination of the ocean is dependent on a certain amount of decay from death. I mean, that's what I saw on the and so that was my question. If it was very good.
Okay. Well, that's a good point, but my point by showing that slide was that God has to make the concentration of all the different ions within the so-called salinity had to be perfect. The concentration cannot change, otherwise anything in the ocean would not exist. They wouldn't live. The bacteria would be surviving at all. So at that point, I don't know, death was not, but the point is, God has to make the ocean more or less perfect. before he could add any of the thing he did day 5 into that ocean. But if there was written something in the slide that I haven't seen, we can come back to that.
Yeah, you do that.
No problem.
Yeah, thanks, Tessa.
Sorry, I didn't mean to throw a monkey wrench into
No,
it. I
that's-
was a little confused about that.
No problem.
Good point. Excellent. We'll make note of that. Warren.
Yes, hi,
Dan.
Frank. I am totally with you in all the molecular complexity there about life. I've watched a series by Dr. James Tour on abiogenesis. I don't know if you've ever heard of him.
Oh yes, of course, of course.
So I loved all that. I totally agree with all you're saying there. But I have a question, because to me, in the area of like refuting millions of years for evolution of life and so on, and for saying it happened by chance, there's no question at all. What you're saying here is powerful to refute that. But what about the other areas of science like astrophysics? Have you read any of the material by Dr. Hugh Ross, for example? He's really he's got the reasons to believe, I think it's called, and he's for the long age of the Earth. He doesn't say anything about, I don't think, about evolution. He talks about the other aspects of the universe and so forth. I've seen so many talks on this where they seem to conclude, the real scientists all seem to conclude that, you know, there's so many things about the universe out there beyond the biological world that indicate millions and millions of years. I wonder what you would have to say about that.
Well, I'm not trying to be somebody that I'm not. I'm not an expert on what's going out there in the space. To my point of view, I'm a biologist. I'm a molecular biologist, and I'm a chemist. And I just have to understand how God created an ideal circumstance for all of us to be able to live there. It had to be an earth and a space around it to begin with. What was out there in the space was not interesting at all. The first most complex part was created on the earth and in the atmosphere around the earth. And to my point of view, is that what I'm studying? And I don't want to refute anybody. I just want to pinpoint the idea that everything has to happen within very short time. It had to be the right purity and the right concentration within all the symbiosis, all the interactions that was around the whole earth. And again, the Holy Spirit had to be there together with the Son, with Jesus and the Father, in order to be able to create all these things around the whole earth at the same time. Because the Holy Spirit can be everywhere at the same time. So, I mean, talking about what's going on out there in the space, for me, no interest at all. Because what I'm interested in is what had to happen on the earth and in the surrounding space around the earth. If you see my point.
Yeah.
So I can't see why we need to have understanding of what the space is producing, because that is a very little complexity compared to the complexity on the earth that I've seen, as far as I see. I don't know if I answered the question.
Good point. And the talk by Robert Mann back in June, I think, in terms of understanding complexity that we do see in the basic elements and how the universe functions, is certainly relevant in this context as well. But I want to just follow up. Appreciate Dan Jansen has his hand up. And Warren, are you finished with your question?
Oh, I could go on, but that'll be good.
Yeah.
I want to be fair with
Yeah.
everyone else.
Yeah, this is just the beginning of a very okay, Dan. Dan, you have your hand up. If you can unmute yourself and ask your question or make your comment.
Sorry, are you saying Diana?
No, Dan Jansen, I see, has got his hand up. Asked to unmute. So if you can unmute
He's
And
maybe in
hear me. Otherwise, I think we'll continue. Frank has a lot of material, I know, and but we'll start to have a
Diana
question afterwards.
Diana has her hand up as well.
Oh, I'm sorry, Diana. All right, well, let's go to you, Diana.
Yes, I thank you very much, and it's great. And I'm a six-day young Earth creationist. I belong to a geology club, which is very lay, very pro-evolution. And I've done an astronomy kind of GCSE with my son just to try and kind of have a clearer view. Just two things really. One, I was in one of the creation ministry Zooms the other day. There's a geologist called Steph Hirima. And just thank you, Frank, talking about the salinity of the oceans. He's doing quite a lot of quite convincing research. There's salt pillars, very high salt pillars in the ocean beds all over the world, which are quite a problem for evolutionists to explain, which they do by talking about evaporation of water. But anyway, long story short, he thinks that salt comes from the splitting of it's of magmatic, from the magma when the Earth crust kind of split apart at the flood, so that the point being the oceans weren't as salty on day 3 and are now a lot more salty with these salt pillars. It's quite complicated. I won't go into it, other than that you've got creatures like salmon that can kind of live in fresh water and salt water. So perhaps it wasn't quite so necessary for there to be an exact salinity at that point. Perhaps creatures were better perfected. But that was my first point. And then the second point, actually going back to what the last question about the James Webb Telescope. I mean, we can't get round that it's kind of transmitting pictures 13.6 billion years galaxies back there. So really, we kind of have to have a bit of a working hypothesis about how to explain that. And my understanding, I bow to other people here, the creation is kind of talk about the speed of time, or God creating things with an appearance of old age, or time.
Yeah, I think that Adam had to see the stars, and all of them, and he had to see it immediately. Why not?
Yeah.
Of course, God created the light between the stars and the galaxies and Adam.
Yeah.
Why not? I don't understand why this is a problem at all. It's just some lights that have been poured out about this story. Forget it. The light between Adam and the stars. For me, that.
It's a big mind shift, but important. Other questions? I perhaps should let you continue, Frank, if and keep your question.
It's good discussion. I would like to go on. I think into something important related to the Earth inside and so on. But I'm not a geologist, so again, all the geology part we have to discuss with the geologist.
This is excellent. Well, let's continue, Frank. And there's lots to discuss. It's just the beginning of a conversation that covers many disciplines.
Yeah. So we still have half an hour to go, don't we?
Yep, according to my clock. Yeah.
Okay, so I can show something more even, or?
Yes, please, go ahead.
Okay, I'll show something more then.
Yeah. We'll continue till the sun sets up there.
Okay, so again, back to the, can you see it?
Yeah, that's good.
Can you see the amino acids there again, the nucleic acids there?
Yeah.
Yes. Yeah, good. So again, back to the understanding of when you have a laboratory. All of you know about PCR, don't you? Have you heard about PCR? Probably most of you have heard about in this SARS-CoV-2 days of the pandemic. Everybody has undergone their test for PCR. And PCR is a very simple work to be done in the lab. It's one of the most simple things to be done in a laboratory. Still, that one is hampered by a lot of different kinds of building blocks. And you need 4 of these here. you need A, G, T, and C. I mean, thymine, cytosine, guanine, adenine. And in order to conduct the PCR, you need the pure conditions of this, and it have to have this right concentration, and it have to follow the right protocol in order to be able to test if you have the virus on board in the laboratory, in the samples that you have provided when you took the samples from your mouth and your nose. So the PCR is a method that needed the same thing here. So again, pointing back to the laboratory, saying that nothing in the laboratory can be done anyhow within millions of years or within dirty compounds, whatever. So it has to happen very quickly in the lab, as quickly as possible, and you need a person to do it, and you need you know, all these protocols and all this arrangement and management. So again, as we said, the whole form of the current interesting way of building up the membrane around all the organelles inside the cells is also something that cannot happen within short time. It had to happen as quickly as possible. You need all these compounds, and how on earth is that going to happen? And you can see on figure 1 here, which is not a very good figure. It's too detailed, but I just show you the complexity is enormous just to build up a membrane around a small little bacteria, and the bacteria is, you know, 1 micron is 0.001 millimeters in size. So how on earth can that be done? Well, again, God did a fantastic work here. And again, how on earth are you going to build? Here we are coming to another compound that has to be done, and it's very, very, very important, involved in the energy consumption, and the energy part of the body and inside the cells has to happen. And again, you cannot get this kind of thing without having the right enzymes, and maybe thousands of different enzymes on board in the cycles of the biochemistry. So again, this is something of a process that has to happen within a short time. And no, we are not even close to the protozoa. We are just talking about some small building blocks here on how that is going to be done. So you see. So again, all of you have seen this back at the time of the school, and you've seen how many of these we need, but I'm not coming into this. Let's go on. So again, the complexity of what we have seen of so many different sciences and so many kind of biological diversity, symbiosis, and plant ecology is so many studies that is coming up, and we need hundreds of new universities in order to start studying this. Please, can you see that?
Yeah.
That the complexity out there is how on earth are you going to study soil science? And it had to happen very quickly, and it had to happen within short time. And today we only know a little bit of it. You know, so maybe 2% of what's going on in the earth, in the soil.
Right.
And how to study it further? I mean, why don't we know it yet? It's because of evolution theory. It's because of the bad ideas, bad ideology, and because of millions of years. This is hampering us so enormously in our studies, in our stuff. Hanging on to the biblical background is opening up all the doors to so many possibilities now to really start understanding biological diversity, really understanding microbiology. And instead of doing that, we are hampered by all the ideas of millions of years, and this is closing the doors to revelation, and the revelation is completely needed in this area. And again, in the terms of talking about nitrogen cycles, for instance, it's not simple, it's complex. The complexity that's going on within a cell that is able to acquire all these kind of compounds within a cell is so enormously complex. And again, the point of view is saying that stars is interesting. It's not interesting. Everything going on in this space is not interesting. What's going on that is possible to be seeing inside the cell is complex. And coming to complexity here, you start to see the cycles that's going on, the so-called biochemical cycles, which most of you have studied in the school. You can see how the complexity is there. And to produce the backbone of the building blocks of DNA and RNA, of course, had to be in place with the right energy consumptions and the right cycles to be happening. And again, it had to happen within seconds and minutes. And then if you start to building up something, what is going on? I mean, if you want to make to put these building blocks together, how on earth is that happening? Of course, it happened within milliseconds. It happened within tens of thousands of seconds. It's just going on. So again, when we go back to the protozoa, we want to make the first protein. What do you need to make the first proteins? Okay, how to make the glucagon? The glucagon protein is in the middle there. You can see it. The glucagon protein is extremely complex. So on earth are you going to make that? I mean, all these compounds that you need in order to make the glucagon, they are not staying alive for more than seconds if you are outside of a biological compound or a biological cell. It's not possible to see it. So outside of the histolytica, the first protozoa, it's not possible to see anything. So you need, in order to start producing this protein, you need first 30S RNA. And if you look at the complexity there, where on earth is that possible to happen with a gradual process within millions of years? It's completely unscientifically to talk about it. Just even talk about it is just impossible. So for us to do this kind of process in the lab, if we want to synthesize or to replace, we can't do it yet. We have to use the cell as it is. We have to grow the cells. We have to grow the bacteria in order to do it. We can't do it on the bench, on the lab. We can't do it yet. You have millions of science around the world, but none of them can do the translation process of a protein. It has to happen inside a cell. Confirming again, God created within 6 days, not a minute more. And you know, the complexity of the 50S RNA, which is completely dependent upon the process, and you need to add the building blocks by the tRNA. And what is coming up? A protein structure. And the protein structure is not simple. Even this glucagon is not very simple, is it? I mean, how on earth, you can't read this, can you? None of you can read this. But what does it mean? It's just a number of letters. Yep, but these complex letters put together in the process of translation. And of course, it happens within seconds, and it has to happen within seconds today as well within each cell. And again, you need a messenger RNA in order to build up the protein. So where on earth is this mRNA and all the building blocks put together? Here is 11. You need 1,100 nucleic acid building blocks in order to create the glucagon. So you need this reading frame here in order to create this protein structure. And of course, it has to happen within seconds and minutes. And by the way, just to make it a little bit more complex, you also need a pre-mRNA before you have the real mRNA, in particular in this first so-called ancestor cell, which of course is not. And the complexity of what you need is just to see in some of the proteins that you need to make the pre-mRNA before you make the mRNA, before you can read it out into a glucagon. And then just to make the chapter a little bit more complex, when you want to make this glucagon protein, what do you need? Yeah, you need chaperones. Chaperones inside the so-called simple histolytic protein. And these proteins, are they simple? No, they are extremely complex. And they work in a complex way, extremely complex way. I'm not going into it. They need, in order to form a functional protein, you need to have this chaperone in place inside the cell. And that again has to happen within seconds and not within weeks or a long time. So you see the complexity of the folding process of making a functional protein or a functional enzyme has to be in place, and not only one protein, but you need thousands, tens of thousands of proteins within the cells in order to have that going on. So if the chaperones is not folding proteins in the right way, the proteins will not work, will not be functional. And then if we start talking about the complexity of a chaperone, we pick out some small parts of that protein. That's just one of the proteins involved. I think there are 6, 7 parts of the protein. Here we are showing one of them. GRU-EL is one of them. And you can't even read it out yourself. This is the building blocks of the chaperone that is completely needed in order to fold and make functional proteins. Okay? And then when we start looking at the organelles inside the cells, now we are starting looking at the so-called first ancestor cell. What on earth is this? Is this a galaxy? This is much more complex than a galaxy. This is just a small little part of a cell. It's much more complex. Forget the stars and the galaxies. This is much more complex. This is the endoplasmatic reticulum, which is very much dependent, and it's just outside of the nuclei. And it have to be there. It have to be functional. And there are millions and millions of ribosomes and other complexities that I've not talked about at all that had to be in place. So again, seconds and minutes have to happen.
Yeah.
So then in addition, you need another organelle inside, which is called the Golgi apparatus, which is also again extremely complex and have to be involved. But I don't have time to go into all this. But as you see, it's involving a lot of transport and transportations inside the cell. And again, seconds and minutes have to be involved, not a long time, not a gradual process. And you need a trafficking inside where you have an enormous amount of highways inside the cells that have to be there, and not one single highway. You need to transport millions of different highways inside a complex cell. And everything has to be in work at the same time within the same cell. Okay? And how to build that road, how to build that highway is not simple.
Yes.
It involves enormously complex structures that have to build into, this is the structure of the highway. And if you put that microtubulus, this highway roads that is within a human cell, well, here you see the picture of it. That's a very interesting traffic system, huh? And to say that that is happening gradually in millions of years, it's completely ridiculous. This is something that God had to put in place at once with the hard work.
Yeah. I'm going to pause for a moment because I do have Philip with his hand up and Dan still. But I'll go with Philip first, if you might comment, Philip, or ask your question.
Oh, sure. I've heard a lot about macro or fine tuning from the astronomers.
Yeah.
Now I'm hearing for the first time from the microscope or from the people that work. Would you call this? Would you sum this up as the macro fine tuning of our creation that cannot be an accident, like neither can the macro? But the macro fine tuning sort of summarize your work here.
Erik Jensen would know my answer here even. No, what is fine tuning? Where does that word come from? I hate it. What is it?
It means that things have to be just right.
It was just right. It was just right in the beginning. It was perfect from the beginning. God made it perfect. It's good enough. And there's no need for any fine tuning at all. So I don't understand the word fine tuning.
That'll be a separate conversation then.
I'm sorry, I may be a little rude here, but you know, fine tuning of what? I don't
physical constants and all of that, I think, is what it is.
But I understand it's a big discussion going on there about fine tunings of whatever it is after the creation. I don't understand it. I'm sorry, because God made it perfect. So perfect, so fantastic, that it's just a word called devolution. That is the word, devolution. There is no evolution, there's always devolution, to my point of view. But again, Philip, is me. I'm talking as a professor here, I'm talking as a scientist, and my theory is my background. You can continue with all kinds of discussions around me here. I'm just presenting the thing that I think that had to be in place in the process of what I've seen within these 6 days.
Yeah.
Very impressive. Yes.
Yeah,
Thank
I think there's a lot. Yeah, and this is, again, I say the beginning of a very important discussion. Dan, are you able to unmute yourself? I see your hand up.
Yeah, I'm here.
Okay, Dan, you had your hand up, so go for it.
Yeah, great. A few comments here. I've been involved in creation science since I was in my youth, and so I noticed Diane Fortune commented on the seeing supernova explosions coming from distant stars, and there was some research done by Barry Satterfield that might be of interest.
Yep.
Interest is a potential explanation for that. That the starlight maybe have traveled a lot faster in its inception when it was first created, because we don't really know exactly what governs the speed of light, and we don't know for sure that it's always been a constant. So it's an assumption. The other thing is that Hugh Ross and all these people that I know of, they all want to bring death before the fall. I think that's a really, if that's a theological issue, it's fairly clear from a reading of Genesis that the first death actually came from the fall, unless you want to talk about microbes or something like that in the gut of humans or something like that. But there doesn't really seem to be any need geologically to go along with the geologic column and have a long age for the Earth. I don't see the evidence for it. The flood explains the geologic column very adequately, except for maybe the top part, which may have been affected by ice age and other more recent catastrophic events. So I'm sticking with the no death before the fall, because I think that's what the Bible says. And so anyway, those are just a couple comments from my side.
Right. And yeah, no good. I noticed a comment, Scott, that what Frank is now presenting is a micro version of what the physicists call the anthropic principle, but that's a whole other set of discussions. And I did actually, there was a comment from Harold, if I was interesting, and it says, it sounds like the problem isn't that 6 days is too short, but that it's too long, since everything has to come together virtually instantaneously. But that's another discussion. In some ways, the 6-day account and process is for our sake, perhaps, in terms of how we were, the creation work was done and how our pattern of life is based on it. But anyway, do you want to comment on that, Frank, in terms, I guess, of the timing and the sequence of things? It's clear the complexity here is incredible beyond what we could even imagine, and it has to all be functioning together as a system is clear. And
but I don't, you know, the protochlorococcus, the bacteria, the cyanobacteria that is producing most of the oxygen around the oceans. I mean, all of them is producing much more than enough of oxygen for the whole world to survive. But but you need all of that had to happen at the same place all over the ocean, all over the Earth. Is that done? It didn't happen just at one place. It happened in the whole ocean at the same time. And that's why you needed the Holy Spirit there to do it all over the place at the same time. And I mean, also in order to secure the balance and to secure the symbiosis everywhere on the earth at the same time. Could only happen instantly within a short time, isn't it? Because otherwise you wouldn't have been able to establish the right conditions in the ocean, in the atmosphere, and in the soil. I don't understand how that can happen without they doing it more or less at the same time. I don't think God continuously fed the atmosphere with oxygen from his own mouth or from heaven. I think he made it come from the photosynthesis, and made it happen around the world at the same time. So I may not have made this clear enough.
Well, I'll collect up other questions you have in the chat, but Frank, you might as well continue. We still have a little time, and be glad for you to complete your presentation or compress it to complete it as it may be. Oh, it's Eric. Yeah, sorry. Yeah, Eric, go ahead.
Yeah. I think there's important that we are not trying to solve all issues with young Earth standpoint at this time.
Yeah.
What Frank is doing is staying within his field of expertise.
Yes.
And that's what he's presenting. So for him to answer questions about death before fall, which is a theological situation, or distant starlight, which is more on the astronomy side, that's beyond his area. And this discussion demands that we talk about different fields at different times. And what he's doing right now is making a very, very excellent presentation within the biological, the necessity of things in place to create biological life at all. And that's not a matter of just that's too long time. The shortness of time is because we're talking about life. The complexity of life itself is far beyond planets out there. And it's not only the initial part, creating it, but it's also sustainability, that this will be a sustainable system that will continue. So I think his presentation is very important in regards to looking within his field, and some of his presentations are outstanding, I would say, in that regard. But that's only my opinion. I'm a good friend of him, so.
Yeah, well said. There's some good comments in the chat, which I'll share afterwards to see how well, and we no doubt will return to this conversation. All right, Frank, I'll let you, if you will, wrap things up. But I think that's well put there, Eric. Well put. Yeah.
You want me to wrap it up?
Yeah.
Yeah. Again, like Eric well said there, and also what was said by Philip, this is from the side of the micro world. This is how I see it as a microbiologist and a molecular biologist. And this is what I observe in the laboratory. This is based on I've done 30 years of hard work in the laboratory, and that's what I've learned from it. And anything outside of that, it's hard for me to discuss in details now. But I'm trying to learn. But the point is that I wanted to open the eyes for the micro world, like Philip said, and to just show you that that there is so many new fields, so many subjects for the research centers and universities and Christians know to go on, that the Christians have to take over the education again, to bring back all these subjects as quickly as possible, to enlighten the universities around and enlighten the research areas, to open their eyes to see that there are hundreds and maybe thousands of new subjects that can evolve and can come out of this. And just to learn from the Bible, the more I read the Bible, the more I learn what we are missing. We're missing so much these days in medicine, in so many subjects. We are missing it because we have a prejudge related to millions of attitude. And the millions of years is just making us completely blind in the scientific world. To my point of view, when you open your door and see how these fantastic things could happen within 6 days and how it's done and how God did this work, then it's opened doors for me to so many interesting fields, so much more that can be done in research. And we have to pull our efforts now together to do that and to see how we can bring back much higher quality into the research and to the field of research and innovation worldwide. Yeah.
Thank you very much, Frank. Very excellent presentation. And again, opened up many areas of further discussion, indeed. Well, thank you. You are involved in a project, at least to develop a science center or research institute at some point. Yes. That's very essential, but also quite a daunting task, I'm sure, in terms of what's required. And do you have colleagues that you're in discussion with that are really getting an idea of what really the implications are of your research? Like, is there a network now that's forming around your work?
That's true. And I will just discuss this presentation that I showed to you in front of a pure secular professor, which is a colleague of mine.
Yeah.
And he's a very advanced professor, and he looks at the same presentation if you have seen, and he had to admit all the time that other theories, all the theories they've heard before, has so much holes. There are so much lacking.
Yeah.
And here we are opening a new world of interest, micro aspects that hopefully we can take on all of us and to push for. Yeah.
Clearly, and the kind of awe and wonder that we see, the more we know, the more we're
exactly.
Utter. I mean, that's amazing, really. So I thank you, Frank, and others for participating. There will be a recording shared with you, and also the chat comments. And then, Frank, if you have any references to a few key papers that might be helpful, I know they're probably some of them quite technical, but if there is any literature you think that would be useful to carry this on, and definitely we'll have to get the astronomers involved in the conversation. That'd be an interesting fit, because I think there's
do you want me to send these papers? I can't send so many of them, but some of them
if you could select a few that you think are really would be, fine.
I will.
I appreciate that.
I send it to you then, and you distribute it.
Yeah, could you everyone then just please put your email in the chat so that I'll have the concise list that I can send that material to. If you'd like it sent, just put your email in the chat box. And any final comments? Again, thank you, Frank, for really opening up a whole, I think, area of conversation and discussion and research that's been suppressed for far too long, and it's really important that this get out. So I'll take note of all the email addresses and that will, I'll be able to send then a copy of the discussion, also papers that Frank would recommend, at least as and then the comments from the chat itself. There's some good comments in the chat. So I'll keep that open and just for you to get all the emails and put that together. Any final comments perhaps then from the group as to perhaps ideas or suggestions that you might have from kind of where to take the discussion from here? Again, as I say, this is really an opening. It's very mind expanding. I mean, just to get an idea of what levels of complexity we're dealing with is really incredible, and I think you've done an excellent job at giving us the big picture on this one, and I really appreciate it, Frank.
Yeah, thank you all for listening. It was a nice moment for me, and I hope we can get this kind of fora.
Yes, I think there's so much there, and appreciate that. And so I will.
Dr. Scott has his name. Dr. Scott Masson.
Yes, Scott. Yes, there you are.
Hi. Very briefly, Frank. So thank you. I'm sorry for joining you late, but I heard enough to make, to my mind, it's clear that it's an impossibility for anything other than a very rapid creation of life everywhere, all at once, more or less. I mean, according to our, but within a very, very brief span of time. Have you written anything at the popular level on this? One of the problems that we have isn't the science, which, as you say, is being neglected and has holes all over the place, but that there aren't things written at a popular level so that people could understand that. Have you written anything on that, Frank? Or can you point us to anything in the area of biology? I know that cosmologists are doing it and geologists, etc., but at the biological level.
I have to be very careful.
Okay.
I don't want to hamper. We have something with Andreas Wieland and the Truth and Transformation going on.
Okay.
I have to be a little bit careful not to hurry, not too much.
Okay.
But it's coming plenty of popular stuff in the years to come. But I have to take it very carefully not to lose my job. And again, it's not because I'm not talking science. I'm talking just science.
Yes, I know you are. Yeah.
But I'm building up a book that is going to be heavy book. But at the same time, Eric and others, I mean, we can come together with a number of people to write more popular scientific papers, also meant for children in the future, that can emphasize this idea of micro world, not on the map, not only stars and all out there.
Yeah.
So yes, the book is coming, and I'm pulling together professors like me to write that book. Even secular professors want to help me write in the same direction, not only Christian. So this idea is coming out of also other kind of understanding that this have to happen rapidly and quickly. So again, so anybody, Scott, if you are interested, please come back and write to me. If you're interested in writing one chapter in the book, please welcome. If you are within this field, I don't know.
I am certainly not in this field.
Okay, sorry. But we can find others who will be in the field. Yes.
Yeah, I mean, I think Michael Denton many years ago, Evolution of Theory and Crisis, went into the complexity of the cell as a point that just was not explicable by any of the current evolutionary theories. But a big project, and we'll look forward to it. Have you got the basic support in place to develop the institute, or is that something you're putting together at this point to develop the kind of a research center that really conducts the kind of work you're recommending?
This is in very good hand, and the process has started.
Good, very good. We'll keep us posted on that.
Yes, thank you.
Okay, well, thank you. We've absorbed a lot to think about, and we'll certainly have a follow-up discussion, I'm sure. I'll bring in some others as well. Thank you for your time, and I will collect all the emails and get the recording sent out in a few days, as well as the chat, and then any articles that Frank would recommend at this stage for us to read. So indeed, we'll pray God's blessing on your work, Frank, and the many that are joined with you. It's a daunting task from the human side, but it's an awesome God that we're serving. So, you're on the right path there. Okay, I will formally close discussion. I'll keep it open just so I can get all the emails in, and look forward to having further discussion. And we'll get others involved as well. Maybe get a small panel together, some key people on these items. All right.
Bye-bye. Thank you, Don, also to you for organizing this and moderating it.
Pleasure.
Thank you so much.
Pleasure.
Thanks, Don.
Yeah, very good. Okay, my mind has exploded right now, so we'll digest this, but it's amazing stuff, and I really appreciate it. It's awesome. All right. I've got I'll keep all that open, and have a good evening or morning, everyone.